Phylogeny is grouping of organisms, okay? When you hear the word phylogeny, we're grouping organisms based on what we're going to term relatedness or common ancestry. So this is an old phylogenetic tree I took a picture of out of another university that shows what's called the tree of life, all right? And there is a website called treeoflife.org that actually is mapping out the tree of life today. And it's scientists all over the planet in all different disciplines that are adding data or information to build the actual tree of life. And so it's a pretty cool website if you ever are bored, which you're not right now, but if you ever get really bored, you can check it out. So treeoflife.org, and it shows you basically combinations of information that'll allow you to focus on if you're interested in arthropod phylogeny or speciation, or mammal speciation, or land plant speciation, or you like fungi, right? It'll allow you to look at the evolution of different organisms and what we know right now, right? And of course, data is always being added to that organization. So I really, really stress these last two lectures on speciation 1 and 2, and on this chapter, which is called phylogeny, to go check out the Campbell books that are in Cell Lounge, and actually look at these chapters to fill in notes if things are fuzzy to you, right? Because a lot of this stuff in the last couple lectures, and this lecture also, are kind of conceptual, right? They're not as hands-on or as not as straightforward. And so you want to make sure that your understanding is your understanding, right? You understand kind of the way speciation has happened, what macroevolution is, what adaptive radiation is, and then how phylogeny works. All right, so we're going to do a lab on phylogeny, which I'm going to be honest with you is not easy. It's a hard lab because phylogeny is not something easy to do. It's going to push your mental capacity as you try to group organisms based on what they look like. It's tedious. It can be very tedious. How's that? That's why most phylogeneticists use computers to let them do all the tedious work, right? But to understand basic phylogeny, we have to kind of go through and understand foundationally how we build them. Now, why is phylogeny important to biology? Because I'm going to be honest with you. When I was an undergrad, I hated phylogeny. When I was a grad student, I didn't like phylogeny. Didn't hate it, just didn't like it. And now, as a professor, I have to teach it and I use it. So it's a tool, right, that you're going to need to at least be able to use or read if you're going to be a biologist or study organisms, take some other classes, for example. All right. So why is classification of anything important? So you notice up here on this slide, I just pulled this slide out of one of my vertebrate anatomy lectures. It says classification of vertebrates. Why do we classify things as biologists? Because like any discipline, you have to be organized. If your notes are chaotic, it's hard to study for your exam. If your notes are really organized, it's easier to study for your exam, right? It's the same with any discipline. It's the same with biology. If I want to study a group of organisms or understand a group of organisms and how they work, then I need to know, well, what's a reptile, or how do I group these organisms? Studying organisms or anything in a smaller group makes it easier to organize thought and find similarities and differences within a group. So I always use this example in vertebrates. There are 20,000 described species of bony fish. That's what an osteichthyon is. If I just threw all 20,000 of those fish in this room, I mean, it would be daunting, wouldn't it? Like, oh my, what would you do with all those? But if we broke them into smaller groups based on similarity, then it might be more fun to study them, right? I can compare this group to that group and so forth. It allows you to actually start making some better inferences on and ask questions if you can observe them in smaller groups. It's almost like taking a reductionist view of how things work, right? Getting it into a simpler way to study it. Of course, comparative anatomist, which I am, and a lot of zoologists, which I also am partly a zoologist, or plant biologist, or anybody that studies bacteria or is a microbiologist, need to group organisms. And the most universal and sound way to do that is through evolutionary relationships, right? Understanding how they're all related to each other, like a family tree. but at an evolutionary level. Now, the first kind of guy or scientist to start doing this was a male, because they did most of science back in the 1700s, was this guy here, Carlos Linnaeus, and he came up with something called binomial nomenclature, which we still use today. This is how we name a species. So if you were to go out and find a new species, say a new species of insect, and you were brought it in, and you can't find any name or anything that looks like it, so you found this new species, you would get to name it, right? And there are rules that you have to follow. But we would give it a two-part name. So every species that's ever been described, no matter whether it's a plant, animal, fungus, protist, bacterium, archaean, whatever it is, gets a two-part name. So, we described 2 million, a little over 2.5, 2.7 million species. Each of them have a unique two-part name. The first part of the name is called the genus. We'll talk about that in a minute. The second part of the name is the unique name. It's probably the first part of the name you would give. The species name has been named for a number of situations. You could name it after a mentor or some scientist that influenced you. You could name it after your kid. You can name it based on what it looks like. So this name is a very unique name. Here are two great examples. Hamiltonii is named after a scientist. Concentricus is named after the concentric rings that you see on that ray. All right? It just depends on the scientist who discovered them and what they want to give the species name, which is the second part of the name. The first part of the name is a genus, all right, or genre, plural. This is a group of species. So, for example, up here we have the manta ray. When that was discovered, it was actually thrown into this genus called manta, because there are several species of large manta ray, which are the largest rays in the world. The same for this species down here, which is a stingray. It's called Urolophus. There are several species of Urolophus other than Concentricus that goes into that particular group. And so a genre is a group of species that have similar characteristics. All right, and it's called a rank. All right, so this is the first or most specific grouping or rank of a number of species. Now notice the two-part name, which is called binomial nomenclature, includes a genus and species. If you were to find a species that didn't fit a genus we have now, you would get to name the genus also. That'd be pretty cool, right? And sometimes if you find a completely new genus, it might not fit into a family either, which is the rank just above it. So you get to name the family also, which happens. But often if you find a spider, it'll fit a genus of spiders, right? And you could place it in there and give it its unique species name. So Carl Linnaeus came up with this binomial nomenclature, and he started naming everything that he could see, and that's kind of what he did. And this sticks. This binomial nomenclature sticks today. Now we take genera and try to group them into similar larger groups just above the genre, which is called the family rank. And so these ranks go up to the most generic rank, which is called a domain, actually. Remember there were 3 domains? Do you remember that? What they were? The eukarya, bacteria, right, and archaea, right, are the 3 major domains, right? So you got here's domain bacteria, here's domain eukarya, here's domain archaea. So if you're a eukaryote, you're in domain eukarya. That's really generic, right? Think of all the eukaryotes there are. Now, if you find a species, all right, so I picked my favorite. This is my absolutely favorite organism on the planet, maybe next to dogs, but are frogs, particularly dart frogs. I've been breeding dart frogs for 20 years. That's an obsession of mine. I have a bunch of them in my office, in tanks if you want to see some. This one I took a long time ago. It's a picture from a species I actually don't have right now. But you can see the genus and species name of this little guy here is called Dendrobates tinctorus. Tinctorus means painted one or painted back. So you can see they're very colorful. They come in yellows, greens, oranges, blues. You think of a color You can find it in a dart frog. These guys have lots of coloration that suggests they're toxic. These guys live on the forest floor and jungles. Dendrobates is one of 3 or 4, I honestly, anywhere from 3 to 12 genera that make up dart frogs. Dendrobates is one of the larger groups. I have a couple different subspecies of Tinctorus, which means mine, for example, has lighter blue legs and comes from a different part of South America, and is called Dendrobates tinctorus patricia. It's actually got a third part to its name. But Dendrobates, this genus, Epibates, Phylogeny, Allobates, there are a bunch of genera of dart frogs fit into one family that includes all dart frogs, which is Dendrobatidae. So this is the next rank up. So all the dart frogs go into Dendrobatidae. You always know a family name because all families end in DAE. So if you see a name DAE, it's a family of something. And so that gives you the rank of family. Now, dart frogs are a type of frog, right? So there are lots of families of frogs, like Lithobates, Bufo, Anaraxis. There are all kinds of families of frogs when you start looking at frogs. So all the families of frogs that make up the entire group or rank of frogs fit into the order Anera. It's a great name, Anera. The nera on the end is tail. A means without. So Anura are tailless amphibians, and all frogs don't have a tail except for one group. And so all the frogs belong in Anura. There are several different orders of species we call amphibians, right? So you got frogs, salamanders, and then a group you probably never heard of, which are called apodans or caecilians. Those 3 orders make up all of the amphibians found on the planet, which is a larger rank, more generic rank, that includes all amphibians, which is a class. So classless amphibia, which means modern amphibians. Amphibians, the class amphibian, belong to a bunch of classes that make up a group called vertebrata, or vertebrata. Some people say vertebrata. So vertebrates have backbones. Frogs have backbones. So do you, right? So you're a vertebrate also. Vertebrates are in the subphylum because there's a slightly larger phylum that is called Chordata or Chordata. And this includes any organism that develops a rod in its back called the notochord. All right? And so vertebrates are one subgroup of that phylum. And then there are lots of phyla of animals, right? There are chordates, there are arachnids, there are arthropods, there are starfish, there are tons and tons of animals. Anything that's an animal belongs in kingdom Animalia, and Animalia belongs in the domain Eukarya because they're all eukaryotes, right? So this is kind of how we group things. Once you get it into a genus that we know of, we can fit it into the rest of them. Does that make sense? This is where most of the controversy happens in taxonomy, is down here. Not everybody always agrees, particularly the family and genus name, where something belongs, right? Now, most of these groupings are based on similarity. They're based on morphologies, what something looks like. That's classical taxonomy. And each one of these, kingdom, phylum, class, order, family, genus, species, is a taxonomic rank, all right, or a level. This is the most specific level, species. This is the most generic level, kingdom, or domain, if you want something that's even more generic. Now, many of the taxonomic groups that are considered classical taxonomic groups are no longer evolutionarily sound. Just because 2 organisms look alike doesn't mean they're related, right? So the reason for that is because of convergent evolution, which we've talked about, and when 2 things that look alike don't always share a common ancestor. So think of dolphins and fish. Dolphins were once considered a fish. Linnaeus grouped them in fishes because they look like fish. They're not, right? They're obviously not. So they're mammals that develop fins because of the water environment, which puts those selection pressures on the limbs. So here's another one. Here's a mammal. There's a species of Panther pardus, and goes from species, genus, family, all the way down, right? The major class here is not amphibia, but it's mammalia. But mammals, amphibians, reptiles, and birds are considered, well, not birds, but are considered vertebrates. Birds are vertebrates, sorry, I was thinking of mammals. So there would all be in that subphylum Vertebrata. I could have used a plant here, but I'm a vertebrate biologist, so I like to use vertebrates. So this is what we call taxonomy. Now, not all taxonomy is evolutionarily sound, so we're trying to fix that, and it's not easy to do. The one thing that you might have noticed on this last slide is there's a subphylum here. You see that? So there are bunches of smaller subgroups under the major ranks. There are supraphyla, infraphyla, subphyla. I mean, it gets really ridiculous. And the reason they're adding all of these subcategories is because they have to break up a phylum if it's not evolutionarily sound. They have to give it a new group name. And so instead of giving it a new phylum, they give it an infraphylum. So you can start, or a subphylum or a supraphyllum. You can start breaking these down into larger or smaller subcategories, and it gets really tedious and really, really obnoxious, to be honest with you. Again, I told you I wasn't a fan of taxonomy and phylogeny in general, but taxonomy in particular. Dr. Milne would be horrified. He's a taxonomist. So in any case, I like it enough now to stay on top of it. So how does phylogeny differ from taxonomy? So phylogeny wants to base things on evolutionary relationships, how closely related something is, two species are to each other. All right, and we're going to do that using phylogenetic trees, or what are called evolutionary trees. And these are dichotomous branching trees that you can converge down onto a common ancestor. The closer two species are, the more closely related they are, right? And eventually, hopefully, you can map out the entire tree of life if you have all the ancestors and all the fossils, which we don't, right? Down to the representative life that actually first arose on the planet. We're going to talk about how you make a phylogeny and how things are grouped. There we're going to be what are called nodes. Nodes equal a branch point, right? So the node is the branch point. And the node, as you're going to see, don't worry, we have slides on this. The node is going to represent the ancestor, the common ancestor. The internode is the branch between nodes. All right, so the length of branch, and that is going to be, they can be short or long or intermediate, which would represent either evolution events, right, or length of time, or genetic difference. Depends on how you build your tree. So the internodes are the branches or lines between the nodes or branches. Who built the first phylogenetic tree? He traveled on the Beagle. The first tree's from 1859. You already forgot all that stuff, huh? Darwin, who's the first guy to actually, publish a tree. And I'll just show you it. That's from The Origin of Species. So he was the first one to show these trees. You remember that picture I showed you in his journal where he had this little tree and it says, I think, and he thinks these I can't remember if I think it's shellfish or something that he was looking at. And how they're related to each other. So he's the first one to actually introduce trees that are now used today in phylogeny. So he's the first to publish a phylogenetic tree. Let's go back to this slide here. So here's a make-believe tree to get your sandwich at the end, right? But we're going to talk about nodes or the branch point. So here's an example of a node right here where my pointer is. And then the branches, or the internodes, are the lines, right? So you got nodes, internode, nodes, internode, node, internode, right? So you can follow them back. And so these branch points, where you get a one line diverging into two, is something called the common ancestor, all right? And then we're going to group some characteristics on the internodes that we're going to call apomorphies, or characteristics we're going to use to separate groups. So that's what we're going to learn in the next class. One other thing that I'll talk about, then I'll be done here, is lingering misconceptions about evolution. These trees can be very misleading. This is from a general biology textbook. This is from a non-majors book. This is called the ladder effect, where evolution is basically directional to humans or to animals that we consider superior in their characteristics. So what this very simplified phylogeny shows, that this living animal today gave rise to this other living animal, which gave rise to this living animal, which eventually led to salamanders, right? And this is completely not the way you should view it. This animal and this animal share a common ancestor that's 100 million years old, way back here. Not this animal gave rise to that animal. Does that make sense? It's not that this very simple animal, what we consider simple or primitive or lower, gave rise to the next animal above it. That's what a ladder effect shows. And a lot of times I'll put humans right here at the top, right? Because we're badass, right? As humans. We're going to be on top of a lot of these trees. But we are just one line of evolution in a very large group of organisms called mammals. And yes, we do dominate because of our big brains and thumbs. Brains and thumbs make us dangerous. But each of these animals, I want you to realize, this one, even though it might appear simple, is not less fit than any of the other organisms you see on a tree. A matter of fact, this animal is clearly very fit for the niche that it occupies. It occupies that niche, right? So is this animal, which is the most complex animal here, on this tree at least, the salamander. It's very obviously complex and very fit as far as the environment that it lives in. So this is a very oversimplified example of how evolution works. We want to see something that's more tree-like, like this, so that that first animal might be here, and that second animal might be here, and then we can trace their common ancestry back, which happened a long time ago, right? Does that make sense? So that the more complicated the tree, the better, the more robust the tree is. So this is what we're going to try to do, is group animals based on common ancestry. What we're going to talk about next is how we do that, right? And it's not easy. Each tree that you see, that you ever see on paper, is a hypothesis. All right, it's not written in stone. No science is, as you guys know. If it's based on true experimental design and science, a lot of what you see is a hypothesis. It would be the best known tree at that point in time. So if I show you a phylogeny of salamanders from 1988 and show you a phylogeny of tree of salamanders today, they look really different. And that one in 88 was great for 1988, but the one today is updated, right? Because of more data, right? The more data, or different type of data that is used to collect trees. Does every scientist agree on which tree is the best? Hell, man, phylogenists, it's a bunch of nerds with pocket protectors that will beat the hell out of each other. It's pretty funny to go watch, actually. I've been to a lot of debates on phylogenies of certain organisms, and those people are pretty passionate about it. So, in any case, but always remember, it's just a hypothesis based on what we know today. So we'll start. This week we're talking about phylogenies. Tomorrow we'll do just some lecture, and then Wednesday we'll lecture. This is the last material for the third exam, believe it or not. And so whatever we start after this lecture will be on the fourth exam. Okay, so which is a good place to stop because we've already had like 3 or 4 lectures. So Thursday we'll do a lab. It's going to be on salamander diversity and phylogenetics and reproductive populations and what a species is. I haven't put that up yet because I'm kind of still reworking how I want the handout to look like. So should have that done here by tomorrow or Wednesday. So I'll get that up when I get it up, probably on Wednesday for the Thursday lab. But I think this is the slide we left off on. We were talking about trees and who were the first to put a tree up, right? That was Darwin. Put the first tree up, first couple trees up. And this is probably the most universal way that biologists nowadays group organisms, is based on what they call phylogeny, or sometimes you'll see the word cladistics. So this word cladistics, the first part of the word is represented by what's called clade, the word clade. And clade is a evolutionarily sound group or taxon. So remember, taxa is a group of animals, right, or a group of plants, or it's a group of organisms. A clade is a specific taxon. It's a group of organisms that are evolutionarily sound, or that you can trace all the way back to a common ancestor. So we're going to define this word in a few minutes on one of the slides. But cladistics or phylogeny is the study of kinship, evolutionary kinship, between either individual species or groups of species or populations. And so this is kind of a representation of a tree. This is how I like trees to look. I'll show you another way that you see trees, but I like the dichotomous branching that a diagram like this shows. And where branch points merge from, right, this is where the numbers are, those are called branch points, or another name they give them is a node. All right, so that's a node or a place where two lines diverge, and this represents the common ancestor on a tree. So you can see here we have 5 common ancestors, right, on this particular phylogenetic tree. And as they branch, that means this common ancestor gave rise to these 2 branches. Branch 1 up here goes to 2, and then the other branch goes down here to taxon G. And this, notice, taxon is kind of separated from the rest of the tree, right? And this one on the bottom, this is called the basal taxon. And often that basal taxon is what's considered an outgroup. All right, again, another word we're going to see here in a minute. An outgroup is a group that doesn't belong to the clade, but is closely related to it. So, for example, taxon G might have some very primitive characteristics or ancestral characteristics that are also seen in the rest of the taxa, but it doesn't have the traits that go into common ancestor 1 and common ancestor 2. All right, so it's going to be missing some of the traits that the rest of this group of organisms has, or what we're calling the clade. So we call this group down here the basal taxon, the outgroup, and then the rest of these, A through F, are in the in group, right? They're actually part of the clade because they have all the characteristics that separates 2 from 1. And so there might be several data points, characteristics, traits that these taxa share that are not found in G. All right, we're going to define what those traits are and what they look like as we go through the lecture. So branch points can be different lengths depending on time of divergence or how much they differ from taxa to taxa. But as you follow the branching, notice it's dichotomous, right? So 2, this common ancestor is going to give rise to 2 branches, which are the common ancestor 3 and the common ancestor 5, right? And if you follow 3, it then branches into taxon A, and then to common ancestor 4, and then B and C, which are called sister taxa because they share the same common ancestor, they have some traits that differ from taxon A. And this is how we're going to move along, and the traits we're going to talk about here are the ones that separate taxa into individual groups. Now notice this one over here looks kind of weird. So number 5, we have these 3 groups. You don't usually see 3, it's 2, right? Dichotomous branching. When you see something like this that has 3, 4, 8 branches of the same length coming from the same common ancestor, what that is called is a polytomy. more than 2, all right, polytomy. And what this represents is we don't know how they differ from the common ancestor and from one another. So this is an unresolved part of the tree that we just don't have either data for, or the data doesn't work well. And so we kind of leave them alone together and really don't talk much about them until we can gather data that will actually support how they're related to each other. So this is another way to represent a phylogeny. So you got a line that goes all the way up, and then you'll have branches that come off of it. I tend to like this less. I like the other type of phylogenetic trees that you see. But your ancestor is going to be below each branch point. So here's the common ancestor that gives rise to these 2 groups and to this group. And so you got ancestral populations at the base of the tree and what we call derived populations or descendants from the common ancestor above the tree. And then again, we'll put tick marks or slashes on the branching lines to represent traits that separate the groups. And we're going to name those, what types of traits we use here on the next slide. So the one thing I always like to remember to point out is that every tree is a hypothesis, right? So it's our best hypothesis or guess based on what we know today. That hypothesis and the tree can be rearranged when new data arises. So what we're trying to do is group organisms in what we call clades. All right, so I mentioned this, and that's where the word cladistics comes from. So we're going to group species, taxon, populations, so that we can trace them all the way back to a common ancestor. So a monophyletic group, or monophyly, which is a clade, is a group of species or taxa that consist of the most recent common ancestor and all of its, wow, that's bad, this word right here, ancestors, should be descendants and all of its descendants. Let's change that right now, then I'll fix that. Jeez. There we go. Should be the most recent common ancestor and all of its descendants. Smaller and more recent clades are subsets of larger ones. So if we do this right, we can trace them all the way back to the kingdom they belong to, right? Like Kingdom Animalia or Kingdom Plantae or whatnot. The larger taxon are older. So kingdom, phylum, class are really old taxonomic and monophyletic groups, if they're monophyletic, and are typically pretty much agreed upon across biologists, or universally accepted. The smaller or more recent clades, like the family, genus, or species, are the ones that are more controversial, or sometimes there's a lot of discussion about, right? Because they are rearranged based on the most recent characteristics. So what do I mean by that? Frogs are frogs, right? We all agree what a frog looks like. But where those frogs belong in relation to each other is highly debated, right? So we can put all the frogs in this room because we know they're frogs and not fish or not dogs or not worms. But then figuring out, all right, these 6 frogs go together and these 6 frogs go together and these 6 frogs go together, depend on how you group them and what characteristics or rules that you use to group them with, right? And not all biologists agree on that. Sometimes it's pretty clear and other times it's not so clear. Welcome to science, right? So that's what a monophyletic group is, or clade, and that's what we're always trying to do. Now, do we always get it right? Of course we don't, right? So we change trees a lot depending on new data. So what are these characters that we're using to define our branches, right? These traits or characters can be all kinds of stuff. They can really be pretty much anything that you want to use that you can get the scientific community to accept that produces a lot of data that you can use, right? So genetics, like sequence, DNA sequence, a great thing to use to build trees. As a matter of fact, it's the most common thing to use now, is the molecular biological data of a DNA sequence. And in particular, they'll use certain DNA that mutates at a certain rate that they know of, like mitochondrial DNA, for example. Or they'll use skull structure, or they'll use bone structure, or they'll count the number of hairs on the femur of a spider. They'll use those types of data sets to build trees. I've used sperm morphology before, all the little parts of sperm, to build phylogenetic trees in reptiles. So it just depends on what traits you want to use or what characteristics you want to use to build your tree. And of course, you can combine data too. Like you could use a molecular tree that was based on DNA and then add skull structure to it to see if the two trees collaborate, are the same. And you can note differences between the two. The more data you use, the more robust the tree is, right? All the trees I'm going to show you are using just a few characteristics. They're really simple. Most people that do phylogenetics or cladistics are going to be using huge data sets, millions of characters. And so they don't build the tree, they plug it all into a supercomputer and let the computer do the work. And even the fastest computers might take months to pop out a tree, right? Because they got to, collaborate, manipulate, and group all those data points together. So there are kind of two major types of traits. Both are important, but one is more important than the other in separating groups. All right, and these are called plesiomorphies and apomorphies. All right, a plesiomorphy. Plesio means old school, right, or ancestral. Morphe means character. So these are primitive or ancestral characters that suggest a distant common ancestor relationship. So in the frog example, all frogs are frogs because they have slimy skin and 4 legs and no tail. Those are all plesiomorphic. They are really good at telling us what a frog is, but they're not really good at separating all the frogs into groups, right? Because all frogs have those characters. So those are what plesiomorphies are. They kind of help us figure out what are the derived characters, the next group of characters, and also kind of help us define our big group that we can work with. Now, a bunch of plesiomorphies together that, for example, define frogs are called symplesiomorphies. Sim meaning same plesiomorphies, or they all share that suite of characters, ancestral characters. An apomorphy, apo means new or derived character, are the actual characters we're going to use to separate groups into smaller and smaller and smaller and smaller groups, or taxa. And we call these derived characters. Shared derived characters are called synapomorphies. And so these are the characters we're going to use to separate our frogs into smaller groups. Right? If we have 10,000 frogs and we separate them into 50 groups of however many, right? Then the separation into those 50 groups are based on characteristics that we consider derived characters. So a derived character is found in a group, but not in the most recent ancestor. Yep.
So is plesiomorphy like more big picture, and the apomorphy is more like specific?
Exactly. Yep, you got it. Now, every once in a while, you'll get a character in a group that's not found in any other group. So you'll have a frog that has a long nose. I'm making that up, by the way. Oh, there is a frog with a tail. How's that? There is one frog species that has a tail as an adult. So it has this unique character that no other frog does, and that's called an autapomorphy. So if you see that word used, autapomorphy, that means it's a very unique character just specific to that one taxon, not shared with any other species. That is a real salamander up there, by the way. This is a man holding it. So this is the largest salamander in the world. That is a Japanese giant salamander. They get about 3.5 to 6 feet long. Live in lakes. The biggest salamander in the U.S. is about like this. Yeah, about like that. And lives here in Indiana, actually. It's just very endangered, hard to find. It's called the hellbender. So outgroup analysis. Outgroup analysis is really important because what we want is what we call a rooted tree. We want a root or a base, a foundation. Because if you don't have a root, then it's really hard to figure out what's derived and what the order of those derived characters are. Now, outgroup analysis can be highly argued because some people may agree and may not agree with you and might have their reasons. But an outgroup is usually a taxon that represents the ancestral conditions and doesn't have any of the apomorphies. Okay, so a great example of this is a little organism that's called the lancelet, right, which is a chordate. You are a chordate. So we're a chordate with a backbone. So for vertebrates, a great outgroup for vertebrate phylogeny is the lancelet because it has all the chordate characters we do, but it doesn't have a backbone. All right, so it shows all chordate characters, but doesn't have the apomorphy that separates vertebrates from the rest of them, and that is the backbone. And it will be your basal taxon to all vertebrates. So all other taxa that are in the tree are called the in-group taxa, or the taxa that make up your clade, all right, which you can trace back to a common ancestor, which is going to sit between the lancelet and the rest of the groups. So here's what it would look like. So here are vertebrates. So notice our tree is rooted with the lancelet. Here's our outgroup. Notice that when you have this common ancestor here, there's a trait right here that is going to separate this bigger group, all the other taxa, from the lancelet. That's the vertebral column. That's the synapomorphy that separates vertebrates from other chordates. Now, obviously, these are all vertebrates. Now we need more characters, don't we? We need more synapomorphies to separate out the rest of the vertebrates. And notice we have 5 vertebrate representatives here. So we're going to use just one trait at a time. If we look at the vertebrates that we have, there is one called the lamprey that doesn't have jaws. It has a round mouth. It doesn't have a movable jaw like we do. And so we're going to use the characteristic hinged jaw to separate lampreys from the rest of our taxa. Okay? So all of these taxa have this synapomorphy, hinged jaw, which separates them from the lancelet and the lamprey. Then we have fish and then 4-legged organisms. So we're going to separate fish from the 4-legged animals by using 4 legs or 4 appendages as our synapomorphy that separates salamanders, turtles, and leopards from fish. And then we'll use the amniotic egg and hair to separate out the rest of our vertebrates. So each one of these traits is a shared derived character or a synapomorphy that separates each of those groups from the others. We can trace them back to a common ancestor, right? If you follow the lines. So this would be the clade vertebrata, right? Or we call it a subphylum vertebrata. All good? All right. So monophyly is a group that you can trace all of the descendants back to one common ancestor. So a monophyletic group or clade, if this is clade 1, would be A, B, and C. Notice you can trace those 3 groups back to here, right? So as long as you can trace all the groups back to a common ancestor, then you are in a monophyletic group. So I can't include D in this group, right? Because I can't trace D to there, now can I? Or I cannot include C, right? C has to be included in the group because C can trace back to the same common ancestor. So if I forget C or try to involve another group that doesn't belong there, then those would be non-natural groupings, right? Or not monophyletic. So the first one I just mentioned Well, let's count monophyletic groups first. Okay, so how many monophyletic groups do you have here? Start from most recent and work backwards to the oldest. So if we do that, we can count. So 1 and 2 is a monophyletic group, right? And 4 and 5 is a monophyletic group. There's 2 groups. And then we could put 1, 2, and 3 together for a bigger monophyletic group that involves an older ancestor, right? And then we can put them all together, all right, and trace all of them back to here. So this, way back in the day, would be a larger taxon like a order or a class, right, or a phylum. These up here would be, like this one here, might be a family, and this might be a genus. Does that make sense? So the bigger taxonomic groups are older. The newer and smaller taxonomic groups, genus and species, or genus in particular, are newer clades or more recent clades, if we do it right. So here we have 4 monophyletic groups, right, that we could count off of this tree. So here is a typical tree. So this is out. You're going to see a lot of trees in textbooks. You take biodiversity or you take vertebrate anatomy with me next semester. We look at lots of trees because it's the easiest way to say, okay, here's where frogs are, and this is how they're related to all the rest of vertebrates. Now we can focus on frogs, right? And so when you draw the tree like this, notice you have one line that starts here and goes all the way up and ends. Typically, would be the common ancestor, or what we would call the rest of fish over here that you don't see on the left. And then there is one group of fish that is kind of our outgroup here, or our basal taxon, that is a really good group as our outgroup because it doesn't have 4 limbs, but it does have lungs, just like all terrestrial vertebrates, or most of them have lungs. And so we know that all the rest of the fish don't have lungs, right? They have gills. And so we're using this as our synapomorphy to separate out tetrapods from the rest of fish, okay? So our outgroup lungfish here are a really weird group. They can breathe air. Isn't that crazy? Matter of fact, lungfish can drown. How weird is that, right? A fish that can drown, really? So if you ever keep one, if you were to, you can buy these sometimes. They're expensive. You have to have a tank where you leave airspace for them to breathe. They have to come up and breathe air. It's really strange. They do have some gills, but just not enough to maintain oxygen concentration, so they have to breathe air. But you can notice that once we get past the lungs, we need to now separate the lungfish from the rest of tetrapods. Well, tetrapod means 4-limbed, so we just use limbs. See that? Our first synapomorphy here that separates our basal group from the rest of tetrapods is limbs. And then we need to now separate the first group out of our tetrapods, which are our amphibians or frogs, salamanders. So we're going to use gas exchanger slimy skin, notice, to separate the frogs from the rest of our tetrapods. Then we'll use the amniotic egg to separate frogs and fish from the rest of our tetrapods. So they all have a shelled or covered egg. So that includes mammals, turtles, snakes, crocodiles, and birds, and so forth as we go up the line until we get to this group here, which is called Archosauria, which includes birds and crocodiles. So all of those groups are monophyletic groups. Yep.
So is this what you would ideally want a phylogenetic tree to look like? Like you would have something separating each group?
Exactly.
That would be
Yep.
the
That's what the branches represent. And here we're only using 1 or maybe 2. You had fur and lactation here, but I mean there could be 200 traits, right? Or 100 here and 500 there, and it just depends on how many characteristics you use. ultimately, if you really do it right, you can only use one trait in the whole tree. If you see limbs twice, then that's going to represent convergence. Remember that convergence? We talked about analogies, which does happen. We'll talk about an example of that in a minute, where the environment shaped the trait, not common ancestry. All right? So convergence becomes a really important part of evolution because it's the environment doing its changes to the organism, not inheriting something from a common ancestor. In this example, we don't have that, right? All of these are used only once. And that's what we really want. We want to, if we do this right, we don't want to add convergence to the tree, but it happens. So convergence is a major kink in our armor here, if you will. So what happens when we don't group them right? And it happens. I mean, it happens. So we might publish a paper, for example, back in the 1970s, they published 70s, 75, 78, something like that. They published a number of papers that said reptiles include these groups. Well, guess what? They didn't include birds. They left birds in their own group. And for a long time, people said that reptiles were reptiles and birds were birds. In reality, once the genetic revolution started and they started looking at gene sequences, birds are reptiles, right? So that original monophyletic group is no longer a clade, right? It's not naturally accepted as a clade because we were forgetting a group, weren't we? We were forgetting birds. Now, I'm a herpetologist and I love birds. I actually photograph birds, but I don't study birds. So all my papers talk about reptiles. I don't talk about birds, so I just ignore them. Because it's convenient for me. That's the way I do it. But a lot of characteristics that birds have are very reptilian and very dinosaur-like. They are direct descendants of dinosaur. They are the modern dinosaur. Birds are. They come directly from the Velociraptor line, which Jurassic Park made famous, right? And that group is called Archosauria, which is mostly an extinct group of dinosaurs. But it also includes crocodiles and birds. Look, crocodiles and birds are actually sister taxon. They're very closely related. It's weird, right? But they share a lot of characteristics. So when we forget a group, like I just talked about with the birds, that's called paraphily. Para means, whoops, I forgot, all right, or a group that's outside that should be part of. So if I say D, E, and F is a monophyletic group, and I ignore G, which notice is sister to F, that's paraphyletic. So herpetologists are really bad at this. We talk about reptiles and forget about birds. In reality, if we really want to do this right, we need to include birds in our discussion. So that's called a paraphyletic grouping. The worst grouping is a polyphyletic grouping, where we say something is part of a monophyletic group, but it doesn't share a common ancestor. So notice here, we can trace D, E, F, and G back to here, and we're including C, and C, you can't trace it there, can you? Right? It won't go to the common ancestor that they all share. Now, if you go far enough back, yeah, sure. If I go far enough back, I'm sharing this common ancestor, but notice then I would be forgetting A and B, wouldn't I? So including C in my discussion, saying they're related to D, E, F, and G is polyphyletic, all right? So polyphyletic means you include species that don't share a common ancestor. Poly means more than one ancestor. This happens with convergence, or what we call analogous structures, which is called homoplasy, all right? So homoplasy is when two organisms look a lot alike, but they don't share a common ancestor. So here's a great example. Here's our mole. Have you guys all seen little moles before? Coming up through your yard, soil, or mulch bed? My dogs used to dig them up all the time, all right? So this is the North American mole. This is the Australian mole. The only real difference is coat color, and it's a little bigger. It's a little bit bigger. But if you look at them, they look a lot alike, right? They're built like little tractors, right? And they have pointed noses, really long, sharp claws, and really strong forelimbs, smaller hind limbs, and a short, stout tail. So if you were to look at them, they have really small eyes. They're really blind. They can't see very well. They both live underground. They dig. That's their job. They dig underground. They spend almost their entire life underground. So the environment has shaped them to look very similar, but they do not share common ancestry. The Australian mole is a marsupial, like a possum or a kangaroo, whereas our mole is placental, like you, right? Where it's a placental mammal, and so they don't share a common ancestor, not a recent common ancestor. So there would be a grouping like this, where we're basically just grouping them based on similarity and not on common ancestry. Okay? We do the same with dolphins and fish, right? Dolphins look a lot like fish. We know dolphins aren't a fish. But have you heard people call dolphins fish? I mean, they do if they're not science people. Because, well, I mean, they do. They look a lot like a fish with their fins and their dorsal fin. And heck, I've seen people call dolphins sharks, right? And they're not. They're a mammal and not anywhere closely related to fish, particularly sharks, which are even older than bony fish. So that's polyphyletic. So this happens a lot when you got convergence. And convergence is hard to discount often. It's hard to, I don't know, it could be, it couldn't be. So a lot of argument goes in when convergence is suspected. So let's look at an example here. If we wanted to build a tree and we wanted to use just the camera eye, right? We would never just use one trait, but here's an example where we're going to try to do this with one trait. And we're going to try to build a tree on the camera eye. And if you look at all the organisms, all the animals that have camera eyes, like our eye that has a pupil that opens and closes, there are obviously fish and reptiles and mammals, but there's also the octopus, right, which isn't a vertebrate. So that's weird, right? It's an invertebrate. And so we want to know, is this homologous? Is the octopus eye homologous to vertebrate eyes? Or is it homoplastic, right? Which means that it is an example of convergence. So if we build two trees, two hypotheses, right? Homoplasy versus homology, here are your two possibilities. If the camera eye is homologous, that means that the earliest ancestor, you got to trace it all the way back past octopi, all of those animals have camera eyes, which if you look at them, they don't, right? Which means that it would have to have been lost by several groups. That would be possible, but probably not probable. The camera eye evolved separately between two groups because of convergent evolution or environmental constraint. That's homoplasy, right? So when you look at the two trees, this is the homologous tree hypothesis. Here's where the camera eye arose. And look at all the groups that would have had to have lost it in order for it to be homologous. It's like 6 different times, right? Throughout the history or evolution of animals, would you have to have lost the camera eye? Now that's a possible hypothesis, right? Homoplasy means that you gained it twice. So 6 versus 2 differences. What do you think is the more probable one? Losing this 6 times or just it arising twice? Arising twice, right? The least number of steps is the most probable, right? We call it parsimony. Keep it simple, stupid, is what my dad used to say, right? So yeah, the least number of steps to get somewhere is the more accepted, or what we call parsimonious tree. So parsimony is a rule that we use in phylogenetics. The least number of steps or assumptions is the most probable solution. Every computer program that builds phylogenetic trees has parsimony built into it, so that the tree the program pumps out is the simplest, most direct tree. Because think about all the possibilities you could get, right? I mean, you could do all kinds of crazy stuff that would involve lots of different changes, which are probably not very probable. So if you look at these two trees where we're putting birds, humans, giraffes, rats, and monkeys together, you have two hypotheses. One has 12 steps and one has 6 steps, right? Notice we put 1 twice here, okay? Which would be convergence when you see the same character twice. But this is the more probable or accepted phylogeny because it's a lot less steps. And that's called parsimony. So where the problem comes in and the confusion comes in is when you try to combine taxonomy, classic taxonomy, where we name things based on what they look like, lots of convergence, lots of problems with it. And we try to use those same names when we do cladistics. And so, when you teach about fish, when I teach vertebrate anatomy, I teach fish. Well, guess what? I don't think about tetrapods when I'm thinking about fish. You don't think about 4-legged fish, right? But we are fish. We are, in a cladistics view, we are the most derived fish. That's weird, isn't it? So fish isn't really a real group, is it? It's not a real, you're not a fish. I'm not a fish. But there are fish, or what we call fish, so it makes it really hard to, what do you call them? Right? But notice if you trace us back, are in the fish, right? We're nested in fish. We're nested in this group here that are called lobe-fin fish. We share a common ancestor. Bet I taught you something new today. Too bad we can't breathe underwater. Wouldn't that be awesome? Man, that'd be awesome. But we can't. So fish is not a real group. It's not a real clade. Dicots are not a real clade. If you were a plant biologist, dicots are anything that have two little leaves that sprout out during their development. And they usually teach, and I teach it when I teach plants because I'm not a plant biologist, but when I have to teach biodiversity, I teach dicots versus monocots. But if you notice, these aren't two monophyletic groups. Monocots are actually a dicot. You see that? They're nested in dicots. They just have lost that cotyledon. At least that's what the genetics suggests. So there really aren't true dicots. Notice there are 2 groups of them. Isn't that weird? So that's a polyphyletic group, right? Down here, this is a paraphyletic grouping, isn't it? Because I'm forgetting us. I'm forgetting tetrapods. So I'm leaving tetrapods out to describe fish, paraphyly. Up here, I'm forgetting about monocots, and this is polyphyly, right? One common ancestor, another common ancestor down here, right? Weird. Prokaryotes are the same way. We now consider prokaryotes as bacteria and archaea. Prior to the 1970s, we thought every prokaryote was a bacteria, right? And that would be polyphyletic, because now we know archaeans are a separate lineage from bacteria, and they share different ancestors. That's the molecular revolution has helped us work those, some of those problems out. Not a monophyletic group, not a monophyletic group, not a monophyletic group, right? But we still use the names, especially when you teach. So, have you ever heard somebody tell you that, I don't believe in evolution, I didn't come from no monkey? They use that really good English too, usually, when they say that. And my answer is, I didn't come from a monkey either, right? I share a common ancestor with hominids, right? That is, we share a really recent common ancestor with the chimpanzees, right? We share 99.9% of our DNA. It's kind of hard to say, it's like that uncle you don't want to talk about, right? I mean, we obviously just recently share a common ancestor, whatever that common ancestor is. But there's a lot of other species between humans and that common ancestor, right? Look up human evolution. Neanderthalis, Hannibalists. There's a whole bunch of different human-like ancestors that diverge on different lines, and now we have just us, right? In the Homo group, so Homo sapiens. And if you trace all of the great apes back, notice that orangutans are our outgroup, right? Or our basal taxon. That is, we have to go way back in time 13 million years or more, depending on who's conservative and who's liberal, to share a common ancestor with the orangutan line. Whereas if you come up here to, this is, 6 million, does that say 6? I don't have my glasses on. 6 million to 3 million years ago, depending on who you read, say we share that common ancestor with the chimp line. So, no, you didn't come from monkeys. You came from a common ancestor of the great apes, particularly the chimpanzee relationship. So we are sister to the chimps, right, in our phylogenetic tree. So here are the chimps. Here is the human, right? So we're separate monophyletic groups that share a recent common ancestor. Okay, phylogenetic bracketing. What does this mean? Phylogeneticists do this a lot. So we have lizards, snakes, birds, and crocodilians. And now I've told you birds are reptiles now, right? Why are birds reptiles? Because they are direct descendants of Saurischian dinosaurs, which include archosaurs. They are modern dinosaurs. I mean, in a simple way. But these dinosaurs, we don't know how they ran. We don't know if they were warm-blooded. We don't know if they had four-chambered hearts, because all we have is bones, right? We don't have DNA. But if you look at the skeletons of these dinosaurs and of birds and of crocodilians, they share a lot of similarity. And so what you do is you take these two groups and you bracket the dinosaurs between them, because crocodilians have four-chambered hearts. So do birds. Thus, most of these dinosaurs probably had four-chambered hearts, right? Because of their ancestry. Birds are warm-blooded. A lot of biologists suggest that Saurischian dinosaurs were warm-blooded, right? Because they're so close to each other. May be true, may not be true. There are other biologists that don't agree with that. Somebody build a time machine so I can go back and see a velociraptor. I would really like to see one. In a cage or something, right? So that's what phylogenetic bracketing is. You put fossilized groups between groups so that you can extract kind of their position and maybe some of their behaviors and soft tissue architecture. Well, I think we ended on this slide yesterday, talking about phylogenetic bracketing, which simply means if you have a good fossil record and you have some organisms that are alive that have characteristics of those extinct forms, then you can bracket them or pull them into the phylogenetic tree. Even though we may not have DNA or organ structure on them or soft tissue structure on them, we tend to make a lot of hypotheses or assumptions that some of the characteristics that you see in the groups, like crocodilians versus birds, both of them share characteristics with a number of dinosaur groups, that these dinosaur groups may have shared some of those behaviors or had similar soft structures like four-chambered hearts and maybe warm-bloodedness, maybe nesting behavior, sexual selection, those types of things. So organisms with really good fossil records, like dinosaurs, are a great example of the ability to bracket. If you don't have fossils, it makes it really hard, right? Another good group, mollusks or bivalves, which make shells, right? Like snails and clams and oysters. They have good fossil records. You can throw them in to bracketing trees on those types of organisms. All right, we also talked a lot about how we transmit genetic information. The most common way we do that is through what's called vertical gene transfer, which is reproduction. So whether that's asexual reproduction or sexual reproduction, mitosis, meiosis, or if we're talking about prokaryotes, if that's binary fission, right? Which is how they kind of replicate their DNA and divide in half. The other major way you can do this, which can cause rapid evolution, is called horizontal gene transfer, which prokaryotes do really well. So archaeans and bacteria, they have, remember, little transportable circular DNA pieces called plasmids that can carry genes, and they can transmit them between two different species. When we're talking about vertical gene transfer, it's from parent to offspring, same species, right? Horizontal gene transfer, two completely different species share genetic information. And prokaryotes do this a lot, and they have done it a lot through their evolution. Eukaryotes can do this also via organs, typically, I'm sure say organs, organelles, like chloroplast and mitochondria. So the endosymbiotic theory is a great example of horizontal gene transfer because each of these, the plastid, the chloroplast, and the mitochondria has its own genome, right? Has its own DNA. So you're not only transmitting the whole organism here, but you're also incorporating its genome into your overall genome within the organism that is doing the symbiosis. So these are kind of the two major ways genetic information is transferred between groups or between species. Okay, lastly, I just remember we can count monophyletic groups by starting with the basic or the most recent branches and then working our way back so that if we do it correctly, everybody on a tree should be able to be traced back to a common ancestor. And so you can count groups, right? The ones on top first, all right, with their 3 different recent common ancestors. And then you got the 2 outer groups, right, that you can trace back to this node, A, B, and C, and F, G, and H. And then you got one more big group here you can trace back to this node, right? And then obviously, if you trace everybody back, it's going to be all the way down to this very old common ancestor, which would be a larger taxon, like a class or order, right? And then you got families and genus, genre maybe, and species, depending on what kind of tree it is. Now, these can be individual species on top, or they could be a genre or genus. They can even be families. You could put family of insects up here, for example. It just depends on what you're interested in comparing and what kind of data set you have for those taxa. Also, I've given you kind of an example of how you could do a phylogenetic tree using traits like wings and color and hair. And then I've given you the answers for that. So what I would do is look at it and try to do it yourself and then compare it to the answer slide. This is a really poor, phylogenetic tree because we have a lot of convergence here, right? Whenever you see traits twice on a tree, that means it's arisen independent of one another. So you got hair and wings in this example show convergent evolution, which is really important in the evolution of a number of organisms because the environment can often really dictate niche specialization, right? And forcing organisms, phenotypes to look similar even though they don't share common ancestry.